Yeah, welcome to another session of Bioinformatics. I am Dr. Y.V. Rajesh, Department of Biotechnology, KL University. In today's session, we will learn about the significance and applications of multiple sequence alignment. So, in previous sessions, we learned about the types of multiple sequence alignments, that is local and global. Then methods of multiple sequence alignments. Specifically, we learnt about dynamic and heuristic approaches with some focus on clustral W or clustral omega here. So, why multiple sequence alignment is done here? So, we know that basically multiple sequence alignment, it is done for with four simple applications here. The first one is phylogenetic tree, second one is homology finding, and third one is motif identification. Majority of the people will do for three approaches: phylogenetic tree construction, or homology finding, or motif identification here. So, basically the idea of sequence alignment is very simple here. So, we learned that DNA and proteins, they are products of evolution, right? So, every living organism on this planet Earth evolved from a common ancestor, that is last universal common ancestor, which is known to have DNA as a genetic element. DNA is a genetic element. It has proteins, cell membranes, it has energy, metabolisms. So, from this one you have bacteria and eukaryotes have been evolved. So, if you take the major or most important enzymes or proteins such as RNA polymerase machineries, ribosomes, or energy metabolism, irrespective of the complexity of the organism. So, the cells have these mechanisms and proteins are conserved. So, the functionality is conserved. That means they may be dissimilar by 80 up to 70%, but 30%, that is 30% of the protein sequences lead to similar function here. So, the idea is very simple. Why the significance of multiple sequence element is very simple. Since DNA and proteins are products of evolution, while comparing sequences here, we can know about conserved sequences, that means which have the traces of evolutions, and non-conserved, which are due to evolutionary products here. So, multiple sequence alignment, it can tell you about evolutionary paths here. So, many applications are there. So, one application is when with pairwise sequence alignment, when your novel or unknown sequence, the function of unknown or novel sequence is found using pairwise sequence, you don't have the confidence in the result. You cannot confidently say that, so the function of my unknown protein is same as the protein which is matching with the database. You cannot have more confidence. But if it is matching with more than one sequence, and if the result is quantified by multiple sequence alignment, you can confidently say that my protein has this novel function here. So, that means extrapolations. So, extrapolation of the results of the findings to tell about the functions of unknown proteins with high confidence, will do that. And second application of multiple sequence alignment, structure finding of unknown proteins here. So we'll learn about structure finding in homology modeling here, and pattern identification is third application of multiple sequence alignment. So you can discover very conserved positions and whether it can be a protein or nucleic acid here, and you can tell that you can tell their importance here. For example, if you take Tata box, the Tata box in promoter regions of eukaryotes is conserved in majority organisms. The TF2D protein of RNA polymerase complex machinery will go and bind and recognize the promoter, and then with the help of RNA polymerase the transcription starts here. So we can identify the conserved positions in your sequences. Similarly, spliced sites, start codons, and protein sorting signals, all these patterns can be identified using multiple sequence alignment here. Similarly, structures. So at the structural level you can identify a protein domain. So it is possible to turn a multiple sequence alignment here into a protein domain identification by constructing the position specific scoring matrix here, and this position specific scoring matrix can tell you about identifications of the domains here. You can use NCBS BLAST and BLAST for getting the deriving the protein PSS for a set of proteins. Similarly, in DNA regulatory elements, the origins of replications, promoters, enhancers, telomeric regions, centromeric regions can be easily identified using multiple sequence alignment. You may use Gibbs sampling techniques to find out these elements here. Just now I told you structure prediction. So when the logic is very simple. So when sequence is matching, sequencers are similar, that means structure will be same or similar, then function may be same. The logic is, so when in multiple sequence alignment, if you found sequence similarity, that indicate that it structurally also same at protein level and function will be same here. So structure prediction, multiple sequence alignment is a part of structure 3D structure prediction here. And another important or most recently or widely used aspect of bioinformatics is SNP analysis. So single nucleotide polymorphisms here. So SNP is non-synonymous single nucleotide polymorphisms, also SNPs, NS-SNP analysis here. So every human being or all the human beings on this planet Earth, they actually same in 99.999%. Same is there. So they differ one nucleotide of 1000 bases here. So one nucleotide of 1000 bases only will have changes among us here. So that one nucleotide change may be lead to some specific disorders or different phenotype is because of this small nucleotide polymorphisms here. So differences amongst the individual nucleotides within the population here. So here let us assume that this is a sequence here. So in individual, one may have T, another may have C, another may have G, another may have A. In a very long sequence here, maybe these small differences may lead to change in the phenotype. Sometimes it may be change in the it may lead to some disease if it is a functional regions or structural regions here. So the multiple sequence alignment has several applications here. So and it is considered as a most important important technology or tool in bioinformatics. So thank you very much for listening to my class. So in today's session we'll learn about applications of bioinformatics. Now we'll learn about